Does cancer loves glucose? Cancer cells have a high demand for glucose, which is a type of sugar, because they rely heavily on it to produce energy through a process called glycolysis. This phenomenon is known as the Warburg effect, named after the scientist Otto Warburg who first described it. There are a few reasons why cancer cells prefer glucose. Rapid energy production. Cancer cells grow and divide at a much faster rate than normal cells. To support their rapid proliferation, they require a large amount of energy. Glucose provides a quick and efficient source of energy through glycolysis, even in the absence of oxygen, anaerobic conditions, which is a common characteristic of tumors. Building blocks for growth. In addition to energy production, glucose is also used by cancer cells to generate the building blocks they need for growth and division. Glucose can be converted into other molecules like nucleotides, amino acids, and lipids, which are essential for synthesizing DNA, proteins, and cell membranes. Adaptation to the environment. Tumors often exist in a microenvironment that is low in oxygen, hypoxic, and nutrient-deprived. In these conditions, cancer cells become more reliant on glucose metabolism to survive and proliferate. They undergo genetic and metabolic adaptations that allow them to thrive under these stressful conditions, including upregulation of glucose transporters on their cell surface to enhance glucose uptake. It's important to note that while cancer cells have a preference for glucose, they can also utilize other fuel sources such as glutamine, fatty acids, and ketone bodies depending on the availability and specific metabolic alterations within the tumor. Understanding the metabolic characteristics of cancer cells, including their reliance on glucose, has been an important area of research in cancer biology. Exploiting these metabolic vulnerabilities is one approach in the development of new cancer therapies, such as targeting specific metabolic pathways or using imaging techniques to detect tumors based on their increased glucose consumption, e.g., PET scans.